Hello, everyone. Thank you for tuning into today's webinar. This is the 19th session of the Industrial Frontier webinar hosted by the Chinese Antibody Society. I'm Ziyang Yang, your host for today's, and also a proud volunteer of the Society. I'm really excited to be here with all of you today. So first things first, let me tell you a bit about the Chinese Antibody Society. Chinese Antibody Society is a nonprofit global organization dedicated to supporting researchers, professionals, and companies in the world of therapeutic antibodies. We're all about advancing the science and making connections. So we host lots of events from our annual conference to the Map Talk Symposium. So all the information presented in today's seminar belongs to the speaker, not to CAS. Speaking of advancing science, we even have our own official journal, Antibody Therapeutics. It's an open access, peer-reviewed journal published by Oxford University Press. If you are doing research in this space, we'd love for you to send us your work. We're always looking for exciting contributions. Now on to today's seminar. We are happy today to have Dr. Shun Zhou from Cyagen Bioscience with us. He's going to give a talk titled Accelerating Antibody Drug Discovery with Fully Human Antibody Mouse, HUGO-Ab, and high-throughput single B-cell screening. Sounds pretty cutting edge, right? So a quick introduction to Dr. Zhou. Dr. Zhou earned his Ph.D. in molecular biology and genetics from Shanghai Institute for Biological Sciences, Chinese Academy of Sciences. He then did a postdoc at Tufts University in the US. With over 10 years of research and development experience, his work spans molecular biology, cell biology, genetics, and more. He's published over 10 papers in journals like PNAS and Acta Biomaterialia. Currently, Dr. Zhou is the R&D Director at the Cyagen Bioscience, where he's leading projects on gene editing, cell and gene therapy, and antibody discovery. He's also involved in several preclinical projects related to antibody drugs. So, all right, without further delay, let's get to the presentation. During and after the presentation, you can enter your questions either using the chat function of the Zoom or into the WeChat group. After the talk, we'll select a few questions from the audience. Dr. Zhou will be happy to answer them. So Dr. Zhou, please take it away.
Okay. Thank you. So let me share my screen. Thanks so much for the Chinese Antibody Association to organization this webinar. So I'm Shun Zhou, so from Cyagen, and now so today's my presentation will be about a fully human antibody mouse, HUGO-Ab, and we partnered with Biointron's single B-cell screening technology AbDrop to provide a one-stop fully human antibody screening service. Our Cyagen, Cyagen is a life science CRO. It was founded in 18 years ago. So we focus on the genetically engineering of mouse and rat models and mouse breeding characterization, and we based the mouse rat models. We provide downstream preclinical studies. So we headquartered in Santa Clara, California, with over more than 900 employees, and we have office and facilities in China, Guangzhou, Suzhou, Beijing, and Japan. So we provide one-stop solutions for researchers And from the research model solutions, we to model analysis and management, we based on our models, we provide drug preclinical studies, CRO services. So we have customers the mouse generation service, and we also have a list of off-the-shelf mouse models. Yeah. So next time, so it is my presentation. So my presentation will cover 3 topics. So topic 1, so why is the transgenic human antibody mouse important for antibody drug development? Topic 2, what are the advantages of a human antibody mouse, HUGO-Ab? So topic 3, so how we can accelerate antibody discovery with the high-throughput single-B technology AbDrop in HUGO-Ab mice. As we know, so actually, nowadays, so more than 100 antibody drugs are approved in the world. So since the first approval of the antibody drug in 1986, actually the antibody drug so increased. So since the 2015, so you can see here, so the trends so increased a lot from the 2015, and currently, so the antibody drug so actually approved for not only for cancer and actually for curing about the immune-mediated disorders, infections, disease, even more the cardiovascular disease and the metabolic disorders and the neurological disorders. So this is because the antibody drug show a lot of advantages. So because of its higher specificity, versatility, and reduced side effect, to make it so a powerful option for treating a wide array of diseases. Yeah. So if we take a look at the top 10 antibody drugs, so in the list in the 2026 last year, so we will see that actually more than around 70% of the top 10 antibody drugs are fully human antibodies. So why is that? So this is actually because all of this you can see the antibody drugs are selling in the MNC, so the big pharmaceutical companies. So why they are prefer to use a fully human antibody? So this is because it is a dominant trend. So they are using the fully human antibody. So next I want to give you some explanation why they are prefer to use the fully human antibody. So as you know, so the first antibody drug OKT3, so actually it is a mouse-derived antibody, but it was withdrawn from the market in 2006 due to the immunogenicity issues. So because it is a mouse antibody, so when it is injected in the human body, so the human body will recognize the antibody and induce the immune response and the triggering production of the human anti-mouse antibodies. And mouse-derived antibodies have a short half-life and resulting in the rapid clearance. And in this case, you need to inject more and more antibodies. So repeated high doses of drug further increase HAMA production. And in some rare cases, so when you repeated injection of the high dose of the mouse antibodies, and it could lead to severe allergic reaction, and sometimes it will result in the patient death. So in this situation, so afterwards, so scientists utilize a multiple method to humanize the antibodies. So they make the chimeric antibodies by fusing the mouse variable antibody region with the human IgG. But actually, the chimeric antibody now is not used anymore due to the immunogenicity issues still. So because the variable area still mouse, and so afterwards they use the humanized antibody by grafting CDR regions onto the human frameworks. So this can significantly reduce the immunogenicity. So in the antibody, and because of the low cost and typically and traditional, so actually in the past decades, so a lot of approved antibodies are humanized antibodies because of the low cost. But actually, it is still bring a risk of the immunogenicity actually. So this is why. So now there is more and more MNC biotechs and scientists are preferred to use fully human antibodies. So actually, there are at least 3 method to generate a fully human antibody. The first way is using the human phage display libraries. So this is very good, but it is but it is only can generate the antibodies with light heavy chain, not naturally paired. Because you do not need to immunize the mouse. So this method is good for toxic antigens sometimes. But actually, because of the absence of the light heavy chain pairing, so sometimes it can still induce the low of moderate immunogenicity. So some scientists will turn to the other ways. The second way is using the single B cell selection from the human PBMC. Actually, this way is very significant. So important for the COVID-19 neutralization antibodies generation, because you can just get the human antibodies from the PBMC, and it's very fast. But its advantages is it can generate the human antibody, but disadvantage is that it is limited just to infectious disease. Actually, as you know, so actually you want to generate antibodies not only for the infectious disease, but also for cancer, for immune disease or cardiovascular. So how to do that next? So we will use the transgenic human antibody mouse. So by using the transgenic human antibody mouse, it is very suitable for most situations. You can use it for not only for single B, for phage display, yeast display, but as well as hybridoma technologies. Okay, so but currently nowadays, so most of the people want to use it, but the limitation is that actually in the market, so the cost is pretty high, and you also need to pay the license terms actually. So sometimes it will influence your decision to use it. Anyway, so I want to draw a brief summary. What is the advantage of using transgenic human antibody mouse? So first is low risk. It is brought the low risk of the immunogenicity for the antibodies. Second is it is efficient, so to screening for the antibodies, because when you're using the transgenic human antibody mouse, it is compatible with the hybridoma, phage, and yeast display, as well as the single B cell screening, so without limitations. And because the antibodies are the natural molecules and derived from the in vivo, so in evolution and selection, so it is more natural. And the 3 is it can save your time, because the antibodies is a human antibody, so you do not need to do the humanization engineering anymore, and so can save you and at least 3 to 4 months actually, and of course save your cost. And because you do not need to do the humanization engineering, so you have no risk to lose your antibodies affinity and developability. So it will be much more important for you to do CMC so process in the future. So with this change and with this so request from the market. So our Cyagen, so is a company to make the transgenic mouse models. We have made the HUGO-Ab. So it is the humanized genomic oscillator for antibody development. So the HUGO-Ab actually is a series product. So we in this series portfolio, so we provide HUGO-Map, HUGO-Lite, and HUGO-Nano. So the HUGO-Map, so it is a product, a mouse product for monoclonal antibody screening. HUGO-Lite, it is a product for common light chain antibody screening. And HUGO-Nano, it is a product for human nanobody screening. Yeah. So my next slide, so my presentation will focus on the three products. And so here is the HUGO-Map. So HUGO-Map, so it is for human monoclonal antibody screening. So in this mouse, so we exchange the mouse heavy chain, copper light chain, and the lambda light chain variable gene area. So with the fully length of human VH, V kappa, and V lambda here. So in this HUGO-Map mouse product, so it contains a full length of all the human VH, V kappa, and V lambda gem lines. And this HUGO-Map, it is generated using Cyagen's turbo knockout ES targeting without using any CRISPR technology. So it's a CRISPR-free. So there's no limitation to use it. So it is a ES targeting technology, very traditional method. And this HUGO-Map will be available in C57, BBC, and SGL strains. So to meet different requests for antibody screening. To validate our HUGO-Map antibody mouse, so we collect B cells from the spleen, and we also collect the serum from the mouse and test the B cell development and the IgG secretion. So compared to the C57 wild type mouse, you can see that the B cell development was observed normally. Yeah, IgG secretion compared to the wild type, it is also comparable to the wild type as well. And we also want to check if the human, so the genes we targeted to the mouse genome is normal, because the VDJ need to be rearranged to form the normal antibodies. So in the mouse, so we collect the B cells and send to the NGS sequencing and using the RNA-seq. And we also collect the data and analyze data to see if the V gene, D gene, and J genes on the heavy chain are rearranged normally. So here we can see that in the B cells, so the antibody sequence contained in this contains the V gene, D gene, and J genes, and we also calculated the gene arrangement frequency and compare to the natural human frequency. You can see that the V gene, D gene, and J genes, so rearrangement frequency pattern is very similar to natural human. And we also observed that the CDR3 length frequency in the heavy chain, so it's very similar to the natural human body. Here the blue curve is the mouse, the pink and the red, so the HUGO-Ab and human. And similarly, we also checked the V gene, V and the J germline genes from the copper light chain and the lambda light chain. So the result is very similar to the human patterns. So they show very, very similar pattern for the V gene and the J gene rearrangement. So the overall, so that means our HUGO-Map mouse is successfully genetically generated, can form the antibodies. And the most important thing, the next step, we validated the immune response capability in our HUGO-Map. So we utilize the different antigens. So from the different target A, B, C, D are from the four different targets. So we injected the antigens in the HUGO-Map, and we measured the titer, the antibody titer in the serum, and we can observe that actually, so each of these case show very high titer of the immune response actually, and the target D is the PDA1. So we use this case as our internal R&D to validate our HUGO-Maps to generate the antibodies. And also we observed that actually, so in the four case studies, the HUGO-Ab are exhibiting slightly very similar, very similar. So immune response compared to the wild type actually. For some case studies, for some individuals, we observed a very slightly stronger immune response. And here is showing the PDA1 case study. So after we immunized the HUGO-Map and we collected the B cells, we utilized the two different ways. One is the phage display, the other one is the hybridoma to do the screening of the anti-PDA1 antibodies. And we also we sequenced the heavy chain, light chain from the B cells and did the blast to check if the antibody, the variable genes are the humans. So yes, they are. Yeah, they are. And we also analyzed the diversity of the heavy chain, light chain. So we can see that actually all of these sequences are with the diverse regions and the regions. So from in the copper chain and so light chain and the heavy chain. And finally, we identified 3 candidates with the two method A2. So the molecule A2 is generated from the hybridoma. C3 and G9 are generated from the phage display. And all of these, the 3 molecules are showing compare. So slightly stronger affinity than the benchmark concentric. And here is showing the blocking acid actually also showing the comparable or better affinity than the concentric. Here is showing another target. We use the B7H3 antigen, so immunize the HUGO-Map, and we also obtained 2 more good molecules compared to the benchmark as well. And next steps, we validated the molecules with the cell-based and animal-based assay to further validate that our HUGO-Map can generate the antibodies with the bioactivity. Actually, from this data, so we conclude that the candidate molecule G9 is isolated from this HUGO-Map mice, so demonstrated enhanced affinity and biological activity compared to the concentric. Here is showing the HUGO-Map's advantages compared to the product in the market. Here is showing the comparison. So our HUGO-Map compared to the other different products. So you can see that actually, our HUGO-Ab, it is generated with the ES targeting, in situ ES targeting technology, and it will be available in the BBC, HGL, and the C57 background. And the most important thing is that our HUGO-Map contains the complete heavy chain repertoire, complete couplet chain repertoire, and the complete lambda chain repertoire. So the diversity, so theoretically, is the most diverse, highest, yeah, compared to the competitor products. And it is the CRISPR-free technology. So it is good. And the next, so I want to give, so why we partner with our Biointron, because Biointron is a company focused on the antibody discovery and antibody recombinant production. But our Cyagen is a company focusing on the mouse model generation and the development. So we two partner together to provide a one-stop solution for fully human antibody screening services. Yeah. Here's showing the, so I want to talk about a little bit about the different technologies for antibody discovery, actually. So here is showing the different technologies, hybridoma, phage display, and a single B-cell technology. As we know, actually, in the past decades, so the hybridoma and the phage display are utilized for the antibody discovery, especially the hybridoma is very typical. But actually, you will know that the diversity of the hybridoma technology actually is very limited, because when you do the screening, so when you generate the plate with the B cells, actually it is very limited, the diversity. It is very label dependent. And when using the hybridoma, yeah, so it is typical. You can get the affinity very good, but actually it is most time cost dependent, and cost at least 3 to 4 months to get the molecules you want. But with the phage display, so you can get a high diversity, but the affinity, it depends. Sometimes you can get a high, sometimes it depends. So because the molecules, the light chain, the heavy chain is not paired, so you can see that actually, so sometimes the molecules you get, so it is still with the high risk of the immunogenicity and the time, and you can save some time compared to hybridoma. But when you're using the single B-cell technology, it can combine the advantages of the hybridoma and the phage display, because it has high diversity, and it can save time actually. So, and the most important thing is it can provide you the paired light and heavy chain antibodies. Yeah. So it combines the advantages of the hybridoma and the phage display. So in this case, so the Biointron developed a micro droplet based single B cells. So it exhibited a very high advantages compared to the other single B cell platforms, because so in these technologies it utilize a micro droplet chip to generate the micro droplet. So in this micro droplet, it encapsulates the IgGs and the antigens and the secondary antibodies. So in this micro droplet, so when the B cells, that single B cells, so secrete the IgGs, and then it form the complex with the secondary antibody and the antigens, and the secondary antigens and the antigens and the secondary antibody are conjugated with a different so groups. So when they form the complex, and then induce the Fredholm fluorescence, and in this case the machine, so use the machine can sort the positive droplets, and then so demulsify the micro droplets and then to do the 10x sequencing to get the heavy chain and the light chain sequences, and then you can do the recombinant expression of the antibodies and do the screening and the confirmation of the affinity. So with this technology, so you can do the screening of millions of plasma B cells in one day and NGS sequencing of the single B cell in 1 week, and obtaining hundreds of naturally paired heavy chain and light chain at one time, and from screening to candidate just 1 month. So next I will show you a few of case studies. Yeah. So here is showing the single, so the case studies with the single B cells in our HUGO-Ab. And so we immunize the anti-PD-L1, so the PD-L1 antigens, and HUGO-427. So is the HUGO-Ab. Now here is the wild type. You can see that. So when we do the sorting of the micro droplets, so here is showing the result and the figure showing the signals in the micro droplet. And finally we isolated the pairs of the antibodies. So from the HUGO-Ab, so more than 1,400, and from the wild type it is 1,200. And finally we can get the unique pairs from the selected pairs, and the 400 for the HUGO-Ab, and 140 pairs for the wild type. Yeah. And here is showing, so after we get the antibody molecules and we recombinantly express them, so all of this, not the 20 of the molecules from each, and then you can see that we get, so in this, the molecules with the EC50 value, so less than benchmark, highlighted in red, so you can see all of this. And we also finally get the antibodies from them. And actually, so you can see actually from these expressed molecules, so more than around, so 80% of the positive molecules are obtained actually. And here we use the cell-based assay and to confirm the affinity and the blocking affinity. So finally we get the 15 and 70 antibody sequences show good affinity in HUGO-Ab and the wild type. But finally we got the 6 versus 6 candidate molecules from the HUGO-Ab and wild type. Yeah, surpass the benchmark in binding and blocking activity. So this case study actually showing that when you're using the AbDrop combined with our HUGO-Ab, you can get the antibodies very efficiently. Here is showing the diversity. So when using the HUGO-Ab and wild type, and you can see the result actually the HUGO-Ab and wild type exhibit relatively abundant VDJ gene usage frequencies. So in these case studies, yeah. And here is a very interesting thing that I want to talk about you, because we use the AI model to test the humanization level. So of these antibodies sequences. So first we established an AI model. So in this AI model, so we trained the model using the data from the OAS database. So because in this database, so it has the camel, human, mouse, rabbit, and rat, and monkey, monkey antibody sequences, and then we train this model. So this is the antibody from the human. This is the antibody sequence from the mouse, and then trained a lot of times, and then the model can recognize. So what is the human antibody sequence? What is the mouse antibody sequence? And then we tested the sequence using the PD-L1 antibodies generated from HUGO-Ab. So here is showing the sequence from the scoring. So you can see that it is very low for the wild type. Here is showing the humanized the antibody sequences. So in approved, here is the approved PD-L1 sequence. So fully human. Here is the sequence from the HUGO-Ab, and you can see that actually the HUGO-Ab and the fully human sequence are scored very high, but the humanized and the white is low. And of course, this tool is free to use. So in our Cyagen's website, if you are interested to use it, yeah. And case 2 is showing another case study. So using our HUGO-Ab, and we also use the AbDrop to do the screening of the antibodies of VEGF. So antibody, and also we obtained the very high affinity antibody molecules from this case study. Here's showing the AbDrop case study 3. So actually, this is very tough target because the homology between the mouse and human is more than 90%. When we do the immunization, actually the titer is low. And at first, so we used the hybridoma to do the screening of these antibodies, but actually we just obtained so only 2 binders. But next step so in parallel we use the AbDrop platform to do the screening of the antibodies, and finally we got 28 binders from this case study. So this case indicated that when you're using the AbDrop, so the single B screening, so actually for those difficult targets, so it is more easy to obtain more binders than the hybridoma technologies. Okay, next I want to go to the second product. So it is the HUGO-Lite. So the HUGO-Lite actually, so as I told you, so it's a product used for common light chain antibody screening. So why you want to use the common light chain? Because actually the bispecific antibody development and more preferred by some scientists and biotech companies because they want to generate the bispecific antibodies using the common light chain antibodies. So in this common light chain antibodies, because the light chain sequence are identical, but the heavy chain are different. But the heavy chain, when you form them together, so you can use the technology knob into hole. So because you can make a mutation so that the heavy chain, the heavy chain, so from 2 antibodies can form together always. But because the light chain sequences are identical, so when you produce this common light chain antibodies in the stable cell line, so in CHO cells, so it will be much easier to do the purification, and the yield of the common light chain antibodies so will be much higher, and still because it can maintain the FAB, so the activity, the bioactivity and the stability, it will be higher and be better than the other formats. So in this case, so we generated the common light chain antibodies. So we choose the top eight VCAP genes to generate 8 different common light chain and antibody models. So here is showing the list. So we generate 1, 2, 3, 4, 5, 6, 7, 8, the single V kappa gene, so caramelized chain. So that means in each product, so it contains only one kappa V genes, and we also generate a mixed four genes caramelized chain mouse product. So in this case, so you can choose or you can pick so which one you are interested in, and then generate the antibodies. So in this case, all of these antibodies generated in one product. In future, you can easily form or generate so bispecific or triple specific antibodies. So anyway, yeah. Here is showing the case study. So we use the caramelized chain 1-39. So combined with the single B, the Biointron's AbDrop platform, and generate the antibodies against the VEGF. So you can see here that in this HUGO light product, we also generated 400 pairs of antibodies and unique pairs. We got more than around like 200 molecules. Yeah. And here is showing the so we use the sequencing to confirm. So if the antibody light chain sequence are identical, so actually so we confirm that it is still the 1-39. Sorry, here is 39, is a typo here. And we pick the sequence, sequence from and recombinantly express the sequence, and we also confirmed the affinity of these antibodies. Actually, the affinity of these molecules are pretty high. Yeah. Next is the HUGO Nano, the third product in our HUGO-Ab so portfolio. So HUGO Nano, so that means in this HUGO Nano mouse, so the variable area, it is a single domain. So without light chain, actually just the VHH. So variable. So the variable area is just a single domain. So generated from the heavy chain of the antibody. Yeah. So because we deleted the mouse light chain, and also we deleted the CH1 area in the heavy light chain, and also we did some mutation in the heavy chain. So the heavy chain just can in this HUGO Nano mouse can form single domain heavy chain antibodies. Yeah, heavy chain antibodies. Yeah. Here is showing so a case study. We generated antibodies with our HUGO Nano mass. We immunized the HUGO Nano, and of course, actually, so we observed a slightly weak immune response actually compared to the wild type. This is scientifically understandable because actually because diversity, because we don't have the light chain in this HUGO mouse actually. Diversity of the antibodies in this HUGO Nano is reduced. So the immune response actually is reduced. So it is understandable. And we collected the B cells, and we also sequence the VHH antibody sequence and confirm that they are still humanized, and they are actually indeed the VHH sequence. And here is showing the diversity of the HUGO Nano. sequence. Okay, so we are still doing some more case studies, and in future, I think we will have more and more case studies. And of course, if you are interested in any of the product, you can contact us, and we are very open to do the collaboration with you at the current stage. And here is showing advantage, a summary for the advantages if you collaboration with us. So we can generate the human antibody directly for you, and no string attached. That means if you collaborate with us, so we just charge you one time. So without milestone, without a royalty, so fees, so will be charged for you. So because we partner with Biointron, so we can provide you the very efficient and powerful, no matter how tough, how difficult your target. Okay, so we can screen the antibody molecules for you. So what you want, and it is very fast because as you know, so when you do we do the screening, so we can finish the screening experiments just in 1 month. Yeah. Okay, so at last, so if you are interested, so please record the email address and send the email to us. Okay, so you can send the email to each of the email box. So one is from Cyagen, one is the Biointron. Okay, thank you. So your questions are welcome. Okay, let me check if you guys have any questions. Okay. Let me check. Yeah.
Thank you, Dr. Zhou, for very wonderful seminar, and it's actually very attractive and very powerful platform. So we have a question in the Q&A box, which is comparing to other platform, what is the cost of your mouse HUGO antibody plus antibody drop service?
Okay. So comparing to other platform, what is the cost of mouse HUGO-Ab and plus AbDrop? So I'm not pretty sure actually if you have consulted or asked a quote from other platform. I can tell you that. So the cost, the price. So when you're using our HUGO-Ab and AbDrop, it's pretty low, pretty low. Here I don't want to talk too much about the exact pricing here because it depends. So what your target? So what is the target and what is the downstream validation or the screening? So if you're very interested in the pricing and the cost, so just please email us. Okay.
Okay.
Yeah, our BD and will give you more very detailed information about the cost. Yeah.
Yeah. So one additional question is that
Yeah.
I'm sure maybe on the market there are some other similar mouse model with the human transgenic. How does your platform compare to those platforms? And not just the cost-wise, but just like in terms of producing high affinity, high selective antibodies.
Okay, so compared to the competitor product in the market, so our HUGO-Ab, So first thing, so our portfolio covers three products: HUGO-Map, HUGO-Lite, and HUGO-Nano. So when you take a look at the HUGO-Map, so our advantage and our highlight thing is that our HUGO-Map contains the full length of the full complete repertoire of the heavy chain, coupled chain, and the lumped light chain. So in the market, there's no other competitor product can contain the full length coupled chain and lumped light chain. Most of them can contain the heavy chain and the coupled chain, but no competitor product can provide the lumped chain. And for the common light chain, so what we provide 8 different common light chain products. You can use it depends on your, so each product. So
common light chain is if it's like 8 separate chains. It's not like a one chain combined on 8 light chain.
Okay. Yeah, separate.
And
from the HUGO Nano, actually in the market, there are not a lot of competitor product actually, as I know. So currently, so from the Nano, so the HUGO Nano is established, engineered based on our HUGO map heavy chain. So the heavy chain is a full length of the human heavy chain sequence and the repertoire. So the diversity is much more higher. Yeah.
Okay, so comparing your like free platforms like HUGO Map, I mean, obviously they have the, let's say, Y-type human heavy chain and light chain, but for the HUGO Light and the Nano, either have only one light chain or has no light chain at all. So what are the affinities of those antibodies producing these 3 different platforms compared to each other? I mean, in response to the same antigen.
Yeah, this is a very good question, actually. So this is why we generate 8 different common light chain products, because actually different targets in different products may generate different antibody, and so maybe so antibody affinities so are different in different products. So sometimes maybe this target A prefers in the common light chain 1-39. The other target maybe prefer to the generate the high affinity antibodies in 3-20. So are different. So we provide this portfolio for our customer to test in the first, and then you can choose. Yeah.
I see. So I think it depends on the antigens to be used, right? We have a question here that is, how efficient does this system work for the GPCR targets?
Okay, so as we know, the GPCR are a tough and difficult target for generate antibodies because it's a 7 transmembrane domain antigens. Anyway, so GPCR is difficult and tough. It is because the antigen, so the protein is hard to obtain, so in vitro. So recombinant expression of the GPCR is not easy. So in this case, so actually in our platform, so combined with our Biointron platform, so we provide a very special immunization method using the mRNA immunization for this specific GPCR target. So after immunization with the mRNA antigen, so the immunization will be much more efficient. And then, so combined, so our HUGO map, because our HUGO map provides very high diversity so of the antibodies, and combined with the Biointron's high throughput screening. So it is very suitable and powerful for GPCR target actually.
Yeah, I guess this maybe can also be extended to other membrane protein that would be hard to express, right?
Yeah.
Okay, so let's see. We have a new question here. So are the class-switched affinity maturation in HUGO mouse similar with those in human?
Here is hard to say scientifically because so we utilize the human genes in the mouse, but actually the immune system, for example, those MHC and DC cells, so the immune system still in the mouse. So we cannot tell. So if the antibody so can generate is very same, identical to the human, but I can say that we are able to identify, isolate those antibody molecules that can match and meet your goal, your project goal. Yeah.
Yeah. For this,
Yeah.
I would think maybe the affinity maturation would be less effective, right? But for the class switch, I guess probably will somehow affected because the class switch is regulated by cytokines like IL-4, interferon gamma. But if you are using the human constant heavy chain, I mean constant region of human chain, then maybe their response to mouse cytokines would be different from that of mouse, right? Yeah.
Yeah, maybe, but you know, so actually isolating and the generation of the antibodies from a human, it's very
Yeah.
limited. So we don't know actually. But according to our experience and those approved antibody drugs, so generation of the antibodies in mouse is doable, is okay, I think.
Yeah, I think in the end, probably the class switch is not that important to us, right?
Yeah.
Because you can always graft the variable region to the constant
Mhm.
region that you desire, right?
Mhm.
Yeah. We have another question is that, we have a new question here. Do you train the AI model by open source or your own data?
Okay, that's good. So actually, so our Cyagen is powered with the AI team. So we have our AI team with more than 30 people. So in our Cyagen company, so we not only use the AI for this antibody. Actually, we use the AI for our animal mouse model generation. So you can check in our science website actually. So we have a database AI equipped with AI. So we call it RDDC. So it's a rare disease. So data center. So equipped with a lot of AI models and AI tools. So we use the AI model. So we establish and code this AI model so by ourselves. Yeah.
Okay.
Of course, maybe we will use some tools open. Yeah.
Yeah.
Mhm.
One question is that in terms of bispecific antibody development, what is the advantage of using common light chain? And the FDA approved bispecific antibody typically use the Fab, both light chain and heavy chain, to bind to antigens. You know what I'm saying? Because you are
Yeah.
suggesting maybe in your HUGO light one, maybe the light chain doesn't contribute much to the antigen binding.
So when you're talking about the bispecific antibody, actually in the world, so I believe that there are more than maybe 100 different formats to form the bispecific antibodies. Yeah.
Yeah.
But when you're using the common light chain, so because in the past 10 years, so not too many people using the common light chain. So because actually uh, so this is because the common light chain mouse are limited to use. I believe that. And and the way you're using the common light chain, so because the antibody still keep the fine. Yeah, I believe that the advantage when you're using the common light chain, that will be much more easier, much more flexible for you to form the bispecific antibodies. Yeah. Mm-hmm.
Yeah, I know there's definitely some antigens that probably mainly relies on the heavy chain. For example, I know that the MP
Yeah.
heptane, that as long as you have the lambda chain there, then it can be bind.
Mm-hmm.
And so, okay, before the end of the talk, maybe we have another question. I mean, which is how long does it take to obtain an antibody with high affinity if using the platform for nanobody screening?
Oh, for nano?
Yeah.
Because for nano antibody, actually, so after you immunize the mouse, so we collect the B cells and then establish the phage display or yeast display. So generally, so the 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 turnaround time is very similar to the phage display or yeast display. Yeah.
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 Okay. Yeah, I think we're at the end of the hour. So if
Mm-hmm.
there are no other questions, let's thank Dr. Shun Zhou for his wonderful talk. And
Yeah, email us. Just email
yeah,
us. Yeah.
okay.
Mm-hmm.
Yeah. So thank you very much. And I would like to thank the volunteers that
Mm-hmm.
made this webinar possible. So I would not read their names. You can just, read their names. Thank you.
Thank you. Thank you very
Yeah.
much. Mhm.